Welcome to your review of gene expression. Many of you have learned about the two-step process of gene expression in prior courses. It is a very important concept. It consists of two actions, transcription and translation, and you definitely need this information to move forward in any class that you are in biology, whether it be anatomy, microbiology, or physiology, or any other biology course. You can see on the screen the different topics that I will cover in this relatively short PowerPoint. Gene expression is a two-step process that we will go through very briefly. In this two-step process, it will lead to heart cells behaving like heart cells, brain cells behaving like brain cells, and it's what makes you different from me. Both of us have roughly 100 trillion cells, and many of those cells are nucleated cells, and in the nucleus, this is where we find our DNA, our human genome. In every cell of my body that's nucleated, it has the same DNA, and yet my heart looks and behaves like a heart, and the neurons that are in my brain look and behave like neurons, and yet they have the same DNA. So what is it that makes the heart look and behave like a heart, a brain look and behave like a brain, and you different from me? It all comes down to gene expression. And the final product of gene expression is shown here in this flow chart is protein. We're going to go from DNA to RNA to protein. DNA is different than RNA, and RNA is different than protein. They look different, and yet these steps are about coding, and the codes change from step to step. They look different. The end result is this structure called a protein that enables a cell to behave and look a certain way. What do proteins do? Maybe we should start with something that's less conceptual. What do they do? In a cell, they have a myriad of roles. I'm just highlighting a few. They can be structural support. They can be enzymes that facilitate reactions that wouldn't necessarily go on their own. They can be receptors where they bind to signaling molecules and affect change within the cell. They can be adhesion molecules where it allows cells, neighboring cells, to link together. This is very notable in places like your stomach and in your kidneys. They can be recognition markers, meaning if someone were to get a blood transfusion, blood from another person, we're worried about the recipient's body recognizing those foreign cells from another person as being self versus non-self. If the recipient's body recognizes these cells from another person as non-self, there can be a transfusion reaction, a huge immunological response, and it could cost the recipients their life. So proteins do a lot of work for us. And the physiology that you might study one day, if you do take human physiology, when you study physiology, it comes down to the molecular level. It is these proteins and what they do in our body. What are proteins? They consist of a monomer, an amino acid. And amino acids are linked together. I like to crochet, and I make blankets. Well, the monomers I use to link together are skeins of yarn. I take these long strands of yarn and I link them together. And I take multiple skeins of yarn to link together. And that eventually creates a blanket, an afghan. Proteins are the same way. They have monomers, things they link together to form a large string of amino acids called a peptide, or even larger, a protein. And you can see here that I have two amino acids, and I'm going to describe the the anatomy of an amino acid, but just show, you can see here highlighted in the yellow that we have an OH group of one amino acid and a hydrogen that's from another amino acid, and those two groups are going to link together. And what does that create? It creates water. Now, water is lost. And when those two amino acids are linked together, it creates a peptide bond. This is called dehydration synthesis. What does it mean when you're dehydrated? It means you've lost water, right? So we lose water when we link two monomers together, and this is called dehydration synthesis. If you want to cut that bond, all you have to do is bring the water back. Water is nature's molecular scissor, if you want to think of it like that. Just bring back the water and you will hydrolyze the bond. So I'm showing here in a little animation type, cartoon type drawing, a series of amino acids linked together with different colors. And as I link them together, I'm creating peptide bonds that you can see in between the amino acids. When we have a single strand of amino acids linked together, as shown here at the bottom, that's called the protein's primary structure, just linking amino acids together. We have in our cells, we have 20 amino acids that are our go-to. That's what we use, 20 amino acids. Some are considered essential, and I've circled them here. Essential means you have to get them from your diet, and what's essential changes depending on your age. Some amino acids are essential when you are younger and not so much when you're older. And I don't care for you to memorize these 20 amino acids. I don't want you to memorize their names. I don't want you to memorize their side groups shown here off of this central carbon. I don't want you to do that. What I do want you to pay attention to is that all of these side groups called R groups hanging off this central carbon that you can see here, that's what makes the amino acid different from other amino acids. They all have individual side groups called R groups. And these R groups is what will dictate what the protein can do. It's really important. They're very specific. And again, the amino acids that are considered essential, they need to come from your diet. And depending on how old you are, some amino acids are no longer essential. Histidine, shown here in a hashed kind of circle, is one that transitions between being essential versus non-essential. If an amino acid is non-essential, it means you can actually make it from another amino acid. So you don't have to get it from your diet. Well, if proteins are made up of amino acids that link together, and the primary structure of a protein means the single strand of amino acids linked together, well, what happens to that single strand? That's like me crocheting just a simple chain link out of the yarn. What do I do with it? Well, I can continue working on it. I can fold it around and link adjacent chains. And when we see proteins, the strand of amino acids starting to fold and link together, the protein starts to have unique properties. And scientists have assigned terms for the complexity of folding that the protein undergoes. The protein folding is really important because when the protein folds appropriately, it has function. I can crochet a simple chain of loops, but that has no function. I can make a long chain of loops out of a skein of yarn until the yarn runs out, and what do I have? I just have this long chain of loops. It has no purpose. I have to fold it correctly. I have to link together adjacent loops, and then I might have, I don't know, a pot holder. I might make mittens or something, but I have to link it, and that's the same with proteins. When they are folded appropriately, they have their function. If they unravel, just like an afghan that I crochet, if the yarn comes loose and the chains unravel, what do I have? I just have, I have yarn. That's it. That's all I have. And the protein loses its function, just like the blanket no longer exists. So what are ways that proteins can come unfolded? They can be exposed to heat, high heat, pH, high salt. For example, you're cooking an egg. You crack open the egg, you put it in the hot skillet. The egg white looks translucent, but when it hits the hot skillet, it starts to turn white, right? Kind of an opaque color. That's the heat denaturing the albumin protein in the egg white. So these three things can cause proteins to unravel. My challenge to you is there another way where proteins don't necessarily unravel, but they don't fold appropriately? What is that? Where a protein can be made, but it doesn't fold correctly, and therefore the cell loses an important player. It loses the protein that's supposed to do something for that cell, and the cell may become dysfunctional. What is that? Let's explore. You need to understand that proteins need to fold. Just like my chain of loops, when I'm crocheting a simple chain from a yarn, a string of yarn to make loops, I have to link them together in some way. So I take this simple chain of amino acids and I start to fold it. I start to link it. And we get things like a secondary structure shown up here with the helix. Basically, I can take this strand of loops that I've made when I crochet and twirl it around my finger, right? That would create a helix. Or I can fold it back and forth, like a piece of paper, if you will, when you're trying to fold a map. That's called a beta pleated sheet. Those are secondary structures. And the protein can be folded even more in a more complex state, where you start to get, you can see here, this thing called a disulfide bridge, where you start to get adjacent amino acids that have these unique interactions, and that's now called a tertiary structure. And in greater detail over here, you can see the interactions of adjacent amino acids that are now, when the protein starts to fold, are in closer proximity and they can interact with each other. That is still showing you a tertiary structure. When two independent proteins or more interact with each other, combine, that's called a quaternary structure. The best example I can give you of this is hemoglobin, where it's actually 4 different peptides that come together to form hemoglobin. 4 different proteins. Truly remarkable. This folding is stabilized through a lot of different molecular bonds and interactions. But a lot of them come from those R groups, the side chains of amino acids that were unique for each of the 20 amino acids I showed you previously. Understand this. If amino acids need to interact with each other to make a protein fold appropriately, changing a single amino acid can change a protein's ability to fold, will probably lead to a change in the protein's shape. Its conformation is what we say. And if it can't fold appropriately, this can lead to a loss in the protein's function. Notice here my little cartoon drawing that these 2 amino acids in my previous picture, shown here in pink. Down here in this picture, I've changed it to blue. That's a different amino acid. It has a different R group, and it might not be able to interact with the R group of the other amino acid that it was supposed to interact with. It can't fold correctly. And if that protein can't fold correctly, we lose function. So this brings us then, how do we get a protein? What generates it? Well, a gene, and I'm going to explain what that is, codes for a protein. It is the coding material. A gene is going to determine the sequence of the amino acids that link together to form a protein. And this is going to be a two-step process, and we're going to change the code every step we go. We're going to start with DNA, and I'm going to explain what that is. We're going to end or go to RNA, and then the final end product is protein. Changes in the DNA can result in a change of the coding. Changes in DNA can lead to changes in RNA, which leads to a change in the protein. And that, if we change the protein, we're going to change the structure, its ability to fold, and the ability for it to function. And that can compromise the cell's function as well. So what is the role of a gene? What is a gene? What analogy can I use? I keep saying it's a code. Well, here's a better way to think of it. It's a recipe. And what does if you cook, you follow a recipe. What does a recipe, you open your cookbook, what does it give you in a recipe? Recipes give you two things. They give you the ingredients, and then they also give you the order for adding those ingredients. You can't have one without the other. I can give you a list of ingredients, but you have no idea what you're supposed to make. So those are two important things in the coding of the recipe. So think of a gene as a recipe, and the gene, the code, tells the cell which amino acids to use, that's your ingredient list, and the order to link them with. That's your direction. And if we take a step back and we think about DNA being this code, this recipe, well, let's put it in a bigger perspective. You've heard of the human genome. If I were to take a toothpick and scrape the inside of your cheek and take off some of the cells inside your cheek, I can actually release the DNA from those cells. I can collect the DNA from one cheek cell. That's your genome. You can think of that as your cookbook. If you look in a cookbook, there are different sections in a cookbook. There's a sauces section or pasta section. There might be a protein section. There might be side dishes. There might be hors d'oeuvres. There might be muffin section and cookies section. There are different sections in cookbooks. So these different sections, if we go back to our human genome, you can think of your DNA as your cookbook, and there are different sections in your DNA. We call those chromosomes. You have 23 pairs of them. And then if I look at an individual section of a cookbook, there are many recipes in that section. I can look at a single chromosome from your genome, and I can find an individual recipe in that section. We call that a gene. And then if I read a recipe from a cookbook, how am I reading it? Is it pictures? No, they're words. And words are made up of letters. So a gene is a recipe. And just like a recipe is made up of words with letters, a gene has its own set of letters. We call those nucleotides. And that's the coding, the nucleotides, the sequence of them. They direct the ingredients. They direct the order the ingredients are added together. To go back to biology, nucleotides in a gene dictate which amino acids you use and the order in which you link them together. In our next segment, we will continue on with this concept.